Good day. Today I'm going to present to you our topic from Unit 1 to Unit 3. In Unit 1, we have the science of heredity and hereditary variation. So here we're going to talk about genes. Genetics, it is the scientific study of heredity and hereditary variation. The artificial selection is the selective breeding of organisms to produce offspring. From the word selective breeding, we are the one who will select an organism to other organism and breed them together to produce offspring. A natural selection, this theory was proposed by Charles Darwin, and it says here that individuals with heritable traits that allow them to adapt to their environment are better able to survive and reproduce than those with less adaptive traits. However, Darwin lacked an understanding of the genetic basis of variation and inheritance. So Gregory Johann Mendel published a paper in 1866 showing how traits were passed from generation to generation in pea plants and offering a general model of how traits are inherited. So in 20th century, it became clear heredity and development were dependent on genetic information residing in genes contained in chromosomes, which were contributed to each individual by gametes, the so-called chromosomal theory of inheritance. Now, we all have also the scope of genetics. The first one is the transmission genetics or the classical genetics study. The second one, molecular and biomolecular genetics. The third one is the population and biometrical genetics. Now, moving on. In Unit 2, we're talking about the chromosomes, the basis of heredity and variation. In prokaryote, unicellular organism with a relatively simple cell structure. The best example of prokaryote is the bacteria, since bacteria do not have nucleus or any other structure which are surrounded by membrane. Next, a eukaryote has a compartmentalized cell structure with components bounded by intercellular membranes. Animal cell and plant cell are eukaryotes. In eukaryotic cell, DNA associate with a special class of protein called histone. The complex of DNA and histone proteins is called chromatin. Okay, next, the cell cycle and mitosis. There are two major phases of cell cycle. We have interphase. It is the period between cell division in which the cell grows, develops, and function. The second one is the M phase or the mitotic phase. It is the period of active cell division. We have also the phases of mitosis. We have the prophase, prometaphase, metaphase, anaphase, telophase, and the last one is the cytokinesis. We have in meiosis is distinguished from mitosis by the three events of meiosis 1. Okay, so we have prophase 1, metaphase 1, and the anaphase 1. So let's move on to Unit 3. We have gene segregation and integration. So what is hybridization? It is the mating or crossing of two true breeding varieties. The P generation or the parental generation, it is the true breeding parents. The F1 generation is the first filial generation, or it is the son. F1 generation is the hybrid offspring. And the F2 generation, it is the second filial generation that allow F1 hybrid to self-pollinate. In Mendel experiment on plant hybrids, Mendel's terminology, we have purple flower color as the dominant trait, and white flower color as the recessive trait. So allele alternative version of a genes account for variation in inherited characters. So that is an example of the previous slide. The dominant allele determines the organism's appearance. The recessive allele, has no noticeable effect on the organism's appearance. Punnett square is a handy diagrammatic device for predicting the allele composition of offspring from cross between individuals of known genetic makeup. Take note that we use capital letter to symbolize a dominant allele and a lowercase letter for our recessive allele. Useful genetic of vocabulary. Homozygous, pairs of identical alleles for character, and heterozygous organism that has two different alleles for a gene. Therefore, we distinguish between an organism's appearance or observable traits is called phenotype, and its genetic makeup is genotype. Okay? So, in test cross, it is a breeding an organism of a known genotype with a recessive homozygous and codominance, another variation on dominance relationship between allele. Okay, so that is an example of test cross. And that's the end of our topic.